So let me go. Shall I start?
Yep. I think you have to click the video clip in the bottom.
Okay, there's four is that.
Fourth one. You have to go to the video clips up there.
Okay, got it.
Hello, and welcome to Public Health in Real Life, a channel dedicated to highlighting key global health topics, celebrating unsung heroes in public health, and inspiring the next generation of public health leaders and professionals. So, Chris, building off of the overwhelming response from last week's program on wearables, we are focusing this series on new developments in biotechnology. And today, the topic is cell therapy and gene therapy. So, these therapies, the industry is booming and is at the forefront of innovation. Deloitte reports that as of date, there are over 1,000 cell gene therapy products in phase 1 through 3 clinical trials globally. And experts predict that within 10 years, there will be multiple, almost the word I saw was there will be a tsunami of approvals. And the end goal here is to treat hundreds of thousands of patients around the world, uh, coming up with new fields of research and new ways of treatments to approach some of those diseases for which we have not found a cure yet. So, how do you see this change?
Thank you, Ashwin. Yeah. Yes, gene therapy is a holy grail that is driving transformation for not only patients but also to the science and the industry. Nearly 20 cell and gene therapy products have been approved by FDA, and there are over 900 cell and gene therapy products which are in clinical trials. This is really a great number. However, the unprecedented cost of this technology is a major bottleneck, and it's a baffling problem to the industries, and it is raising concerns to business leaders on sustainability of the business model. The pharma industries are constantly looking for ways to improve supply chain capabilities, reliable manufacturing infrastructure, and highly specialized talent that is needed to improve the access to these technologies and to solving this baffling problem.
Yep. So, as Chris discussed, gene therapy is a game changer for treating a wide variety of diseases, including cancer, cystic fibrosis. Today, there was actually news of the world's first surgery for spina bifida. The surgery was done while in the womb, where cell therapy and gene therapy was used towards ensuring the baby is born without some of those complications associated with spina bifida and other diseases. This research is looking into is the heart disease, diabetes, hemophilia, and AIDS. So today we have Dr. Vibha Jawa. Let's bring Dr. Vibha Jawa here to this screen. Hi, Dr. Vibha Jawa. So
Hello, hi.
So, Dr. Java is an executive director for biotherapeutics, bioanalysis in non-clinical disposition and bioanalysis organization at Bristol Myers Squibb. Vibha is responsible for leading biotherapeutics and cell gene therapeutic bioanalytical function, supporting drug metabolism and pharmacokinetics and immunogenicity, and provides strategic and scientific oversight of the biologics and developmental portfolio. Vibha was at Merck for 4 years, where she led the predictive and clinical immunogenicity group, and at Amgen for 14 years, supporting discovery to development for biotherapeutics. Vibha has over 20 years of experience in diverse fields of biologics, vaccine development, and gene therapy, with successful support of over 20 investigational new drug applications, biologic license applications, and marketing authorization application in the Europe, and in supporting all those successful filings. So Vibha is a recognized leader in bioanalysis and immunogenicity with over 75 peer review publications, and serves as a reviewer and editor for the American Association of Pharmaceutical Sciences Journal and the Journal of Pharmaceutical Sciences. She is an active member of multiple scientific societies and consortiums, especially the International Consortium for Innovation and Quality in Pharmaceutical Development. It's usually called the IQ Consortium, the SC Space Consortium, and the European Immunogenicity Platform. Within the American Association of Pharmaceutical Sciences, she is a steering committee member of the Therapeutic Product Immunogenicity Community. She was a past chair of Immunogenicity Risk Assessment and Mitigation Community, and leads the IQ Consortium for Cell Viral Gene Therapies. Vibha enjoys volunteering as a board member for the State Youth Orchestra and mentoring high school students on STEM related projects in her free time. Dr. Vibha, we are excited to have you on our show.
Thank you so much, and thanks for your kind introduction of me. Thanks.
Thank you. So, to level set our audience, we are hearing about three concepts in the industry: cell therapy, gene therapy, and gene editing. Can you give us a quick overview of these three fields of research?
Sure, yes. And as you mentioned, that there are not even hundreds but thousands of trials in this novel modality field, and the field of biologics has actually moved on or evolved into these more targeted personalized therapies, um, and it is utilizing some of the components of vaccines or even gene editing, which was used more in the research environment, now more translationally to apply to clinic itself. So that led to this is the reason we are seeing a lot more of these modalities, because the initial success has led us to adapt these technologies. So, for example, if we talk about cells, we always knew that the immune system was very capable to kill anything foreign, and being able to harness the same ability to fight the foreign organisms, but in this context, using the tumor cell as your foreign entity was the pioneering work at University of Pennsylvania. And similarly, gene therapies harnessing the ability of a pathogenic virus to be able to go and infect cells very efficiently, but being able to remove those components of the virus which are pathogenic, but still be able to infect any kind of cell, uh, was again a great discovery. And the last one, the gene editing, which just came out, was more about in vivo gene editing using again the mechanisms that a bacteria uses to cleave to stay alive when the bacteriophages are infecting it. So it's amazing that we are able to leverage some of these mechanisms and then apply it to treat diseases.
Wow, that's nice. Thank you for sharing, Vibha. So basically, these therapies are sounding like it's more working at a microscopic level. So especially in the case of gene therapy and cell therapy. So how do they get on to cure diseases at a whole body level?
The whole concept of gene therapy evolved when the human genome was sequenced, and very slowly we started identifying the gene defects that were leading to diseases. And then the next step was, is there a way for us to correct that gene defect instead of treating, as we had done before, the downstream impact of such gene defects, which were the proteins that were abnormal. So, one very good example is hemophilia, which was treated always with providing transfusions of the recombinant factor 8 or factor 9, which were the um, problematic proteins that they were not expressed by the patient well by the genes that were defective, or they had point mutations or spliced mutations. But then the recombinant technology helped us to make those proteins ex vivo and then re-administer them as a protein-based biologic. So now, if we go to the upstream of that, and if there was a way to correct that mutation or even introduce a gene which could make the functional uh, part of that protein itself and replace with the defective one, then you're really curing the disease at that molecular or genetic level, as you mentioned. And that was the concept.
Interesting, Vibha. So, this is, as you're speaking, we can sense that this is pretty advanced regenerative medicine that we are talking about. And from a research and development perspective, um, there are some unique challenges to this field, and especially in terms of like coming up with the types of stem cells to use in terms of the cell therapy, or determining the dosage, that is the quantity of cells to be transplanted. Can you go a bit into these types of challenges for the research and development folks, and how do they tackle some of these challenges?
Sure, yes. So currently, we tackle the therapy or the cellular therapies, which are called the chimeric antigen receptor T cells. We wanted to do the most obvious thing, and that was, are there ways for us to make our T cells, which are efficient killers, much more efficient? Can we armor them in a way that they could go and recognize the tumor specifically and start killing them? And that's where the whole technology around the chimeric antigen receptor T cells was evolved, and now it's led to 4 or 5 very successful filings and approvals. So these are hematological malignancies, and the T cells are available as a raw material from the patient itself, and that's called the autologous therapies. So, and then once they are brought out, then there's a whole process of engineering the right kind of receptor, which would target the tumor antigens. There are next generation of these cars where we are adding some level of armoring, because we want them to be able to also engage a T cell with the effector function, so that there is a very similar to what we have done with dual domain bispecifics or bites. That whole technology has now moved into a T cell, which now is made into a killer, but it's also very targeted to a tumor antigen. So, from the perspective of the current approved therapies, the raw materials are the T cells, and
Right.
what could make it much more efficient, and which is happening already, is the ability to make off the shelf cells rather than using autologous CAR T cells, just because of the length of time it takes from vein to main delivery, when the patient first gives the cells to when we give it back to the give the engineered CAR T cells back to the same patient. So if that could be avoided, the next level is to make off-the-shelf allogeneic cells, which are now cells from maybe all of us, which has been engineered, but we have knocked down the problematic or the sequences which make us non-self for the patient. So like graft versus host disease mechanism reductions, or improving the T cell repertoire specificity to be targeted to tumors. So all those are happening, but they're still happening with the primary cells. So you mentioned the stem cells, and that's a great way. And there are the pluripotent stem cells being used currently to to adapt to the same kind of a concept. And similarly, natural killer cells or gamma delta TCR T cells, these have ability to kill more effectively, but they're also not recognized by the immune system as foreign just because they are the innate phase immune cells. So we still need to do a lot of like knocking down of the graft versus host mechanistic molecules like class 1, class 2 MHCs, or and also introduce some genes so that they can make them much more immune suppressive in the new environment. But these are all evolving technologies with a lot of promise.
Two questions there. So one is about the T cells have to be sensitive and specific to um, the cancerous cells in the body, right? So are those T cells engineered to kind of target those cells that probably, I don't know if it is P53 gene or so how do they recognize those receptors to the cancerous cell?
So that does require first the understanding what are you targeting on a tumor antigen, and then is and similar to how we used to make antibodies, the same concept is applied that you have like some kind of a antibody, and we just take the SCFV region of that antibody, which is the region which will go bind to the tumor antigen and block it, so that it is it targets that tumor antigen, but then it also the T cell is also given some more ammunition to be able to be a better killer because tumors are well known to suppress T cell responses. So if
Hmm.
there is a way for us to engineer into the T cell a much more effective killing mechanism, which could be engaging the cells to become more active in terms of like getting more proficient in killing. So you add that engineered sequence with the tumor antigen, which targeting sequence, which will go and target it. So both of those go hand in hand to make it much more efficient in killing and also targeting specifically.
That is fascinating. My second question, I think almost you answered because I was thinking of asking about like would the body just like it can reject when we do the blood transfusion, it the body rejects some of these transfusions. So similarly, does it reject the T cells as well? But you said that the T cells are engineered to be more to yeah, overcome some of those cancerous cells, uh,
Yeah. So
refusal.
I think the autologous CAR T cell therapy was the reason it's successful is because you're taking the person's own blood,
Yeah,
engineering it ex vivo, and re-administering. The only risks there come from any kind of gene which we are introducing, and when it makes the protein, is that protein now considered foreign to that person?
right.
Yeah, and for that we do a lot of those early developability exercises of making sure that the sequence is as inert as possible, and that we have leveraged our learnings from the therapeutic biologics, but we have done a lot of work to make sure our biologics are inert when they go in, they have the least immunogenicity risk, they have least safety risk. But when we talk about the CAR T cells, there are still risks because one, you're introducing T cells, which are now you have made them very efficient. So when you reintroduce them, even if they're autologous, you could cause like adverse events like cytokine storm, and that has been noted as one of the adverse events that is managed. There are conditioning strategies in the clinic the clinicians use either by giving them upfront or even during the course of dosing, but those are now much more well defined. What we are still there's an uncertainty is that lentiviruses are used to introduce these genes in these T cells, and have we made our the CMC aspects of these CAR T cells, which are our drug products, uh,
Right.
very very much like good specifications, making sure we reduce those CQAs as much as possible, and make sure that there's the minimal amount of these residuals, whether they are adventitious products which are part of the culture or even the gene related aspects, have we reduced those risks so that the T cell has the ability to go in and expand and then be able to go kill the tumor.
It's very interesting. Thank you, Vibha. A follow-up question. Sorry. My next question is, so the input materials, workflows, and manufacturing processes are playing a role in scaling up and decreasing the costs of gene therapy. So, what are the ways to overcome scale-up challenges in gene therapy manufacturing?
Yeah, it's a great question. And, I want to point our audience to this very nice paper which was written, I think this year or maybe last year, and it was talking more about how to make the CAR T cell therapies more affordable for developing countries. But it has a lot of good learnings because it can be adapted even by developed countries. And there are some proposals of how to improve the overall like process itself, and that can help us get away from the manufacturing challenges. And so the growth and expansion of these primary T cell, it has different components. First, you bring them out after when the CAR T cell therapy starts in autologous T cell therapies. It starts with taking out the blood from these subjects and then being able to like go and engineer or do the re-engineering, adding the lentiviral based transduction step to introduce the CAR Ts, and then you have to bring it back. So the vein to vein time could sometimes like around 4 weeks, and you have to know that these are subjects who are critically ill. And the worst part is that by the time you come back with these re-engineered CAR Ts, the subject is not even there because they're so ill. They need that therapy right away. So decentralization of the overall process, because we have seen that there are only some facilities that can do this kind of GMP kind of manufacturing and making sure the quality is maintained. And so the cells are sent to the centralized place, and they do the transductions, they do the necessary expansion of these cells, and then they're shipped back. So one way to reduce that whole step is to be able to do this more on site, which would require infrastructure. But those things are the ones that we need to focus on. Like, can you reduce that time in between? The other thing is expansion, and the CAR T cells need to expand. So we give them a lot of these stimulants which help to expand them in the culture. And again, we can look for the alternatives to do those kind of activations. And it has to be the quality is the main aspect of it, which needs to be maintained. And I mentioned the allogeneic CAR T cells because that will take away the time which we are currently like spending in getting the engineered CAR T cells back to the subject. Because if we have off the shelf allogeneic CAR T cells, then once we know the tumor and the antigen, and then you can make these kind of cells and then just deliver them as any other drug product to the patient.
And we understand that whenever we are providing cell therapy, we do the transfusion-based approach. But when it comes to gene editing and gene therapy, is that the same concept, or how is that administered to the patient?
Yeah, so that's a little bit different. So, it does not make it any less complicated because the challenges are more about making the right kind of the, let's say, AAV. There are so many serotypes, and each one of them have their tropism to the tissue which you are trying to target to.
Mhm.
But there is a whole, the AAV manufacturing has its own hurdles, because again you have to make the vector with as less residuals, and the residuals there mainly come from empty capsids. So making the whole packaged viral vector in sufficient amounts and making sure that all the impurities and the residuals are taken care of, it requires a very well invested infrastructure to do that. And that again has been a bottleneck for viral gene therapies to make these AAV viruses. in bulk amount with a high quality, and also making sure that they can keep those empty capsid amounts low. So it's an art almost. And again, we have seen a lot of good places where it can be done very well, but then for startups or for developing countries, the infrastructure and the training is needed on how to amplify such AAV viral vectors and make them in sufficient quality and amount.
And
Um,
this,
and the CRISPR is again, CRISPR or editing technologies is the another a completely new animal, because there you're trying to do this either in vivo gene editing. So, or even if you want to deliver the whole vehicle, the vehicle like liposomes or nanoparticles,
yeah.
they have to be well characterized. And so we are going back in a way to our like leveraging our small molecule or like mass spectrometric expertise to make sure analytes and metabolites are well characterized, because that's where the problems arise. What are you using as a vehicle? What is their disposition in vivo? Are we sure that we're not going to introduce something new in terms of an entity which will stick around? And all of these gene therapy or cell therapies, or even nucleic acids or CRISPR Cas editing based delivery of gene editing approaches, they also come with a big risk of integration into the genome or mutations or mutagenesis. So there are new kind of risks which are coming up with all of these newer therapies.
Yeah, and especially even when we think about the disease like Alzheimer's or dementia or Parkinson's, they have to go through the blood-brain barrier to make some of those changes, probably. But when we talked about the vehicle, the for cell therapy versus gene therapy, is the vehicle for both blood, or is it for gene therapy and gene editing? Would it be more like a intradermal or intra? How is that addressed?
So currently for the CAR T cells, as we know, the most of the approved CAR T cell therapies are targeted to hematological malignancy. So it is really related to cells coming from our blood, which have been changed and then retransfused back. Uh,
And
but for viral gene therapies, as I mentioned before, the tropism of the virus to a certain tissue does help us to target those viral vectors directly to the organ. So, muscle delivery for muscular diseases, it could be IV, but targeted to muscle, but it could be also targeted to a tissue itself. In the case, as you mentioned, like intrathecal delivery for neurodegenerative diseases. So there are ways for us to focus the delivery on specific tissues based on the tropism of the virus, but it could be done more as a IV delivery also. But hopefully, the tropism of the virus will bring it to that tissue.
great. So so far we covered the bench part of the challenges and developments associated. Would like to now shift the conversation to the bed part of it. So, in terms of bringing it to the patient, the first step I'm sure is the clinical trials. So, when it comes to clinical trials, probably we are looking at uh, it might be a bit challenging because patients with certain diseases that you are targeting might be of low population. So, how do you increase and get enough use relevant data with clinical trials?
Yeah, and this is more of a concern with rare orphan diseases with genetic defects, and that has been a big bane for these kind of therapies, because first of all, um, these gene defects may not be very well that prevalent. So most of the biotechs or biopharmas, they would like to make gene therapies for diseases where they can also get better return. So with a big population. But when you come to the rare orphan disease one, the clinical trials are very limited, because where do you get your patients? And you can see the gene therapy for, let's say, Duchenne muscular dystrophy, and it's challenging because the number of subjects are very less to put into a trial, and as you know, it is a very highly competitive field for drug developers. A lot of people are trying to develop a DMD based gene therapy, but then we are also cannibalizing on our patient population. So, so I think the rare orphan disease population, um, from a regulatory perspective, it is given that designation because there are less subjects. So, but we could leverage a lot of our uh, previous historical experience with such diseases. We have worked on enzyme replacement therapy, so there's a lot of knowledge on how those rare orphan diseases reach the clinic, even with limited set of data. We would also need to like rely a lot more on algorithms or in silico modeling, or being able to do the exposure responses using smaller number of subjects but being able to project. So that has been one way to really understand what that population response would look like. But it does, that challenge does stay. So safety is very imminent. Like a lot of focus is on what is the safe what dose for these subjects, and can you show consistently that there's an impact on efficacy. And then you go with a smaller number of subjects when you're trying to file, but hopefully you can build up that number of subjects. And there's a lot of post marketing commitments, or like registries that the sponsor may have to support, just to be able to continue to monitor for safety and also collect that information over a period of time, and just so that we have enough confidence that these are safe and they are also targeted therapies.
Great, Vibha. So you shared that recruitment is one of the biggest challenges in any clinical trials. So what kind of strategies do you use to increase the recruitment of individuals or participants for the clinical trials? And do you use strategies like decentralized trials or real world evidence? Because you already shared that you use in silico data to some extent. So do you use some real world evidence? And my second question is, so you said post-market surveillance is something that you have to focus to ensure patient safety. So what kind of post-market surveillance practices do you use?
So, to answer your first question about the real world, so the orphan diseases sometimes are really, we don't know much about it. So it's the sponsors learn with the when they dose subjects, and then you collect all the data and keep on mining it as much as possible. Uh, but I think the other aspect of the rare diseases is that you have to work with the patient advocacy groups, because they are the ones who may be more tuned into the patients um, who are coming together in one place, and working with them sometimes does help. University of Pennsylvania, for example, has a very strong patient advocacy group for DMD. They have a whole department which like monitors, manages, and they work with the patient families. And so it's a great resource, because you're able to then understand what is the where is the need, and having those kind of advocacy groups associated with hospitals or clinical trial sites would be one way. The real world data is great, but as you know, some of these therapies are brand new and novel. So sometimes we get lucky with the gene defect and well characterized gene defect, and some of the original trials, like I mentioned, hemophilia, there was a lot done. So there was a lot of knowledge which had been generated already. So that can be now taken and put more into the gene therapy with the knowledge what works for the trial management or even the patient safety. For your second question about post marketing, I think mainly the post marketing surveillance or registries are done to monitor safety long term. And all these gene therapies or cell therapies or CRISPR edited gene therapies, they always have this risk of, would there be a mutation, like the mutagenesis or insertional mutagenesis, and how are we keeping track of that? And so there are these checks and balances in place where um, anything, if there's a overexpression of the CAR T, for example, then it triggers look for insertional mutagenesis, and these assays are very well defined under CLIA so that they can be done to use for the future monitoring. There are some good examples of where and we know that the CAR T, the first CAR T subject which who was given CAR T in UPenn, she just, I think she reached the 10 plus year mark. And so it's showing that it has great remission, and the CAR T cells may persist at very low levels, but what you have to do is see is safety, like there's no other risk due to those CAR T cells except for making sure they're targeted to the tumors.
Wow, that's very interesting. So already you say already there are assays in place to monitor the post-market effects. This is very interesting. Yeah. Thank you, Ashwin.
Okay. Yep. So, Vibha, in terms of speaking still in terms of within the hospital setting, we also after these clinical trials are over and these medicines come to the market, we are heavily dependent on doctors who need to have the scientific knowledge to administer, take care of the patients, and also attend to some of those risks. So, how is the scientific community educating the doctors? Is this happening at the medical school level, or is this happening afterwards?
I don't think we are there at the med school level. Maybe people who are specializing in oncology now may be exposed to it, but we do need very targeted, almost continuing medical education kind of courses or trainings in places where it has been well established. And there are some cancer centers which are very good. They have been doing where like companies are collaborating and then are able to bring the initial findings into more of a commercialization kind of approach. And all the cancer centers where the initial few trials, or even the university academic centers where these technologies were developed and then moved on to the hospitals, that's where there are opportunities for new students or new fellows or new medical students to get trained. And so, but is it formal? Maybe not. It's more about the interest and being part of that group and learning from a scientific perspective. More education is needed, more awareness of what all it takes to bring such a therapy into a patient, and what are the hurdles, and how do you manage the adverse events, and how do you manage a subject post transfusion. So those are good um, tutorials or good learnings that could be done more on a like a classroom basis, or maybe through online tutoring, or similar the way other topics are um, like disseminated. CAR T cells and how to manage a clinical trial of CAR T cells or gene therapies would be a great tutorial.
And in terms of this providing this CAR T cells, how can just want to ask you this question while you were speaking about the cells? How can the just like general public, we give blood to American Red Cross and we donate blood. Is there a way general public can contribute to the research, maybe by donating T cells, or uh, can you educate us on that?
I think yes. When you are going to the off-shelf CAR T cells, which are the cells that I mentioned, could be anyone's cells, but your then we the technology would be there to knock down the class one, class two MHCs, now like knock down the T cell receptors that are specific to that person, but then be more and make it much more inert, and also a more armor to go kill tumors. So those are off the shelf cells, and they are coming from uh, people, peripheral blood mononuclear cells of healthy people. So, yes, there is a way to do it. Now, where would you go donate? I don't know if it is a Red Cross, or maybe we need to like be more, make people more aware of where such kind of collections can be made in a more sterile setting, because these cells are going to be then like they have to be prepped to do the engineering. So definitely, I think that knowledge or that awareness has to be built on where are those centers. And again, the decentralization may help, because currently, if there are only few locations where such
Yeah.
kind of T cells are manufactured, then not everyone is going to be able to provide that kind of donation. But if you have decentralized facilities which are capable of processing, prepping, engineering, then you are going to see a lot more of that population or demographics contributing to that um, facility. And I think for third world countries, you have to be doing that much more, anyways, because there's so many hurdles to process, prep, or even ship, and you may lose the quality of these cells due to just the limitations of an environment where you can't ship them in an efficient way.
Yeah. And it's
That's
good actually you talked about the developing countries, Vibha, and just want to ask a question regarding the pocketbook. So speaking about paying for the treatments, whether it is developing countries where majority of the health is out of pocket expense, and even here we have the Center for Medicaid and Medicare Services paying the majority of the patient bills. Now but I am pretty sure I'm at least imagining that CMS is usually involved in the discussion very late stage. But you know, FDA and some of these other ASPR and other BARDA kind of organizations, they already are involved in much earlier stages of research and development. The CMS kind of folks or insurance companies, they come very late into the discussion, and it the burden falls on the patient. So how can we make it more affordable and bring in more funding streams for the patient's bill to go down?
Yeah, I don't work in that area, but I mean, from a perspective of early development, early manufacturing, reducing making the entire process more efficient, and finding ways to like utilize much more of the AI-based tools to improve the processes. We will have to somehow make this much more affordable in terms of the cost of goods we are using to manufacture these very sophisticated therapies, whether it's gene therapy or cell therapy, or we have to just from our end, like from a developability perspective, we are trying to just be more efficient. Like, how do you make a sterile environment in a smaller space, and is are there bioreactors that could be more, let's not do the way we have been always doing, and I know it worked and that's the success reason of success, but is there a way to move to a more efficient bioreactor to amplify these cells? Are there ways to reduce the residuals or the CMC related questions that may come? And can we upfront start with much more of those processes rather than doing it later on, which adds more time and also adds more prep work? So I think I don't know if I can answer your questions related to affordability because I don't have that know how, but at least what we are trying to do is, because even if I feel that these therapies are always going to stay much more involved, and maybe the off the shelf cells may be much more manageable in terms of cost, because now you're not adding all those steps of taking the blood and, like, processing, sending it out, bringing it back, because it does not only mean time, but also means a lot of like different processes that have to be put into place, and they could all go away if you have the off the shelf PBMCs or CAR T cells which are ready. And so it's almost like a drug product like we use in a emergency room or in a hospital setting. So if we go to that extent, then we're only worried about how do you store such cells and what is their viability, because most of the engineering has been not done in a urgent space. which adds to the cost.
Right. And
That's right.
kudos to you guys, scientists, for thinking about the cost aspect very early in the value chain.
Yes.
Yeah. That's really nice, Vibha. Thank you for sharing. So, from a product development perspective, how long it takes for a product to be developed, a gene or cell therapy product?
Yeah, so it can almost take like a year to develop sometimes a product like this. And I mentioned the vein to vein time, so vein to vein time is 4 weeks, which I said would be not very beneficial for someone who's very sick. But now with these processes which are being implemented, it could be brought down or cut down. And if we can work with off the shelf, then there is no vein to vein time. You give the patient what they need right away once the diagnosis, and we know what kind of tumor and like antigen specific therapy they need.
Sure, that's nice, Vibha. My follow-up question on that is, so cell and gene therapy is available for a range of diseases. Do you think that we will have a few more diseases added to this list in the next upcoming years? That's number one. And from because as you have a vast experience in this field, so is there some strategy to decide whether cell and gene therapy will work for this category of disease but not for the other category?
So one thing we are struggling with is so right now the CAR T cells are for hematological malignancies. We are trying to be able to target solid tumors, and
Mhm.
so there is a little bit of a thought process needed. One, how to deliver um, such therapies, because it's not easy to invade tissues or solid tumors, and you have to change your strategy on how do you want to deliver, or even are you targeting the right kind of the cells in that tumor environment, which is in a solid matrix. So that's one place where the CAR T cells or even engineered T cells like NK cells, or I mentioned the gamma delta TCRs. And so maybe we have to change what cells we are going to use to target such solid tumors. Regarding other diseases, so I know that I saw recently that a CAR T cell therapy could be utilized for treating an autoimmune disease, which we have till now managed, because most of the autoimmune diseases are about management of the inflammatory response of these immune cells that are targeting your own tissues. But there is promise that CAR T cells could help to actually treat lupus, for example.
Wow.
So that's a great application of CAR T cells in a non-oncology space, and it's also a much bigger market in a way for patients because lupus is much more prevalent. Yes, if it's an immune modulatory therapy, and we know there are mechanisms where diseases are caused by those imbalances of immune modulation. So these CAR Ts can work there. It's just that are they affordable enough? Because there are enough small molecule, large molecule biologics which are already there. So how do we compete in that market with making these therapies much more manageable in terms of cost? That then only we can start thinking about applying it to prophylactic disease or even non-oncology disease.
Very insightful. Thank you so much, Vibha. This is wonderful. So let's switch gears now to towards regulation. So you have been in the regulatory space for a long time. So and the regulatory compliance with cell and gene therapy is really a baffling problem, and it needs arduous efforts. So what are the key regulatory challenges for gene and cell therapy products across geographies, and how comfortable have agencies like FDA become?
So we have been having constant discussions on the expectation from a regulatory perspective, and my space has been mainly the preclinical development and like what a package for the first and human dose would look like. And so what are the efforts we need to do from a preclinical developability perspective or a bioanalytical perspective to put the right kind of immune monitoring in place, or making sure that we are detecting what needs to be detected from gene expression perspective. So the guidance isn't draft, but as an industry, we are very actively engaged with the agency by getting feedback and also providing them with some level of like our experiences in an industry setting or a IQ setting. So through didactic sessions with the agencies, the concept is let's work together on it. We know what the challenges are in terms of logistics or even challenges of expressing a fairly good quality of vector, for example, for viral gene therapies, and uh, how are we removing our residuals, or so those are the questions that are in the agency's mind. It's mainly safety. It's either safety because we are using a virus as our one of the vehicles to deliver a gene of interest, so virus on its own, or the gene which is now being given in a someone whom, and how does that get, is it safe, are we doing enough biodistribution studies, is there a risk of viral shedding after the fact, and is that an infectious virus? So all those are the things that the agency wants us to work on and have robust assessments upfront, provide that data at the time of filing, that we have done our due diligence to do long-term monitoring of these subjects and look for the adequate safety signals. And all that goes in the package, whether we're talking about pharmacology or um, like a PK of the drug, which in case of gene therapies is actually gene expression. There's a transgene, and you can look for proteins, but most of the time they're asking how long will this gene expression persist, and is there a risk for it to integrate or cause unnecessary mutagenesis.
Wow, that's really nice. So, a follow-up question on that, as you said, both it involves both preclinical as well as clinical information. So, what IRB looks for in case of cell and gene therapy products?
So I think so. One thing which we found was, and at least is that how much do we know about the safety of the dose of a gene therapy product of CAR T cells? How much dose would we give, and like what is our strategy of dose escalation, or are we looking immediately for some clinical parameters or monitoring for those kind of immune activating signals, because those are the ones that can really derail any trial. So management and those escalation strategies, and using some level of modeling, but maybe also using leveraging the non-clinical information as much as possible. I do have to say that CAR T cells don't have a non-clinical um, models to work with. So we are really relying on some of the ex vivo understanding of interaction of these tumor antigens with the relevant T cells and killing of cells and potency. In the case of viral vectors, there is non-clinical data from some of the animals, but we have also seen that the toxicities or any kind of adverse events that are seen there may not be reflective of a disease state in a subject. So you have to keep that in mind with very gradual dose escalations, but be mindful that these are very sick patients, so you cannot take a whole year or 2 years to do those escalations. So it's risk benefit kind of a management. So the IRBs are looking for all those, what kind of checks and balances are in place. Do you have the right kind of monitoring built in, and are the conditioning regimens also um, like robust enough to manage the adverse event in the setting.
It's really, thank you, Viva. This is really awesome. Yeah. Yeah. Especially when you said already the patient in the ailment, so it's really super challenging to conduct the clinical trials. So this is awesome.
Absolutely. And in terms of Vibha, in terms of ethics, It has the gene therapy and cell therapy. There were initially lots of concerns surrounding ethics around using human cells. So, where is the field now in terms of the ethical stance?
So I think the gene therapy from a viral vector perspective, the concerns were always that are we going to somehow mutate our own human genome by inserting these genes from an external vehicle. And I think with the AAV we have shown that this is not an integrated, like the virus does not integrate. I know that it can persist, and so those are the monitoring that is built in as part of safety. But I think with the success of these gene therapies and the benefits that they're providing, and long-term monitoring not showing any of those effects, um, those concerns are there. But because FDA has been, our regulatory agency has been very good at uh, putting the right kind of checks and balances, there is a little bit more, um, you know, adoption or embracement of this, uh, the whole thought process that gene therapies are good in a way, and they're also treating the disease from that molecular level. Cell therapies, we are a little bit like, yes, you're adding genes into your own cells. So, but at least they're your own cells when it comes to autologous therapies, and even uh, the allogeneic cells, it may be other cells from another person, but you're knocking down most of those, the genes which can cause graft versus host disease. So in the spirit of transplantation, right? Like transplantation was someone else's organ being transplanted into the patient. So that is an acceptable approach. So if we think about it like that, cell to cell therapy, it's still a transplantation. It's just that you're adding some genes to augment that cell's ability to kill tumors or to treat an inflammation. So again, it's the next generation, but as long as the right checks and balances are built in to monitor and also know what the adverse events are, I think it's still a viable approach.
Makes sense.
That's great. Yeah. The next question is, gene therapy has a great impact in transforming patient life. So there are a lot of concerns, on the ethical issues. Initially, there were a lot of concerns, but how is it now? Where is the field now in terms of ethical sense?
Uh, I think, Chris, I think we have covered that question. We can go to the next question.
Okay. Okay, it's okay. It's time to rethink the intellectual property and pricing practices that prevent global access to genetic therapies. How should healthcare systems approach gene therapies to deal with cost and accessibility at a global scale?
Yeah, and I think we covered a little bit of that in terms of how can we streamline each step, starting from like if it is gene therapy, like manufacturability of a viral vector and decentralized versus centralized, like making the looking at each step, maybe using some kind of AI machine learning models to learn from previous experience to reduce wherever we feel that there's redundancy. And but like from a hospital's level, or like how do you make it more, um, I didn't get the last part of your question, but affordable or even accessible. Sorry, can you repeat your question?
Yeah, it's accessibility at a global scale. Yeah.
Yeah, so accessibility has to be by making each uh, like area of a very self-sufficient training is very important, because the technical staff, the operators, the people who are actually going to handle these cells and the quality control around it. So those processes have to be put into place at that level of the hospital itself, because you're not going to rely on a remote entity monitoring all of those practices when you're going to administer these cells or therapies in the local hospital setting. So a lot of like education and training, and that goes back to that specific government of that country, or so at each level, the process has to be supported by overall, like the government regulations, training. The hospitals have to build in new trainings or get their employees trained by sending them where it's being done much more like already has a success, and then bring it back so that you can do it in your own facility.
That's really great, Vibha. So we are coming to the end of the show. Uh, Ashwin, do you have any questions? Yeah, please.
No, thank you, Vibha. We've really learned a lot, and we've seen cell and gene therapy from multiple ends, from starting from the scientific community, research development challenges, manufacturing challenges, and then how we can get it accessible for the patient, and then looking at it from a global health perspective. And it looks like a promising field, and we need the participation of multiple players in the community, including non-profits, advocacy organizations, hospitals, labs, companies, and the government agencies. So it's really a multi-dimensional, multidisciplinary subject, and really fascinating in the way you explained and put laid out things. Was pretty articulate, and we have a better understanding now. We live with better understanding. So thank you for this informed conversation.
Thank you for your insightful questions.
Thank you, Vibha. Really, we are really excited to have you on our show today. So, as Ashwin said, it's really a cornucopia of great information. So much of information you brought from your 2 decades of your research. So, this is really amazing. Yeah. I know you're swamped with so much of work in the week on the weekend, and thank you so much for taking your time.
No
We are really
problem.
super excited.
No problem. Thank you.
Thank you.
Thank you all. Thank you, Vibha. Thank you, Ashwin. Yeah.